Hello everyone, I'm Berta Canal and I'm going to explain you one of the projects of my PhD, which is about male sterility at high temperatures. I'm mainly interested in studying how high temperatures affect reproduction in Drosophila, and as you can imagine, this is important in the climate change context. It is expected an increase of temperatures of 4 degrees by the next century, but also the occurrence of cold and hot extreme events affecting species viability and reproduction. Although the viability range is broader than the fertility range, it is not enough to survive, so species need to reproduce in order to ensure their persistence. It is widely known that some species become sterile at cold and high extreme temperatures, and even though this phenomenon has been documented for some species like Drosophila, it is not well studied. However, some species can recover fertility if they are transferred to mild temperature. And this is what I did in this first experiment. We used the Portuguese Drosophila melanogaster strain, and the main goal is to know if flies grown at high temperatures can reach the fitness of the control flies grown at 25 degrees, or they present a plateau below them and do not improve with time. So for that, larvae were exposed at these 5 different temperatures shown here, ranging from 25 degrees to 31 degrees. And after eclosion, males were separated and transferred to 25 degrees, allowing them to recover. The experiment lasted 10 days, and we measured the number of offspring every day. So every day, the same male was allowed to mate with the virgin female, grown and kept at standard conditions, as we are only interested on the effect of temperatures on males. And after 12 days, we were able to count the offspring produced for each day. So we observed that flies grown at high temperatures did not reach the fitness of control flies, which is this black line here. They produced less offspring throughout the course of the experiment, and they needed more time to recover. And this time is proportional to the temperature. So as higher the temperature, more time is needed, being 3 days at 31 degrees, 2 days at 30 degrees, and 1 day at 29. And you can see that when males are young, they are sterile. We did this experiment not only to get some information about the fertility of this specific strain, but also to know which are the most interesting temperatures to study on further experiments. So we decided to have at the end 5 temperature treatments. First, larvae were exposed at 25, 29, or 31 degrees, but after eclosion, males are divided into 2 groups. So half of the males are allowed to recover, and the other half will stay at the growth temperature. In that way, we can determine the recovery dynamics by comparing these treatments. So from now on, all the experiments will be with these temperature treatments. In order to determine why males are sterile and the subsequent fertility reduction at high temperatures, we suggested that a delay of the accessory gland development is produced. Then, if the accessory glands are not completely developed, they will transfer to the females when mating less seminal fruit proteins, which are essential for an optimum fertility. In this way, males grown at high temperatures should have smaller accessory glands than control males. And this is what we observed. This plot shows accessory gland size of males grown at these different temperature treatments, and we measured accessory gland size on 4 days during sexual maturation to see the growth dynamics over time. We can see that differences arise mainly from day 2 to 6, and it's important to show that focusing on the males grown at 29 degrees, there is no difference between recovering flies and non-recovering flies, indicating that the temperature when adults is not so important in that case. So temperature during the development determines the fate of the accessory gland. Following this direction, if we expect less seminal fluid proteins to be transferred to the female during mating, or the composition of these proteins is altered, the females will be more prone to remating, as seminal fluid proteins reduce females' receptivity. Thus, females mated first with the male grown at high temperatures should show higher remating rates than females mated with control males. For this experiment, we grew flies as before, having two groups of males, recovery versus non-recovery males. When they were 5-day-olds, they were allowed to mate with a virgin female, again grown at standard conditions, for 2 hours, and we measured the behavior, including the duration of copulation and the mating latency, the mating rates, and the offspring number after 12 days. 2 days later, the same female was presented with a stably male, grown at the standard conditions, and they were allowed to mate for 2 hours. So stably mutant males present these non-crossed short bristles, which can be easily differentiated from the wild-type phenotype. And we measured again the behavior, the remating rates, and the offspring number after 12 days. In that way, we can determine by the offspring if the females stored mainly the sperm from the first mating, showing wild-type offspring, or the sperm from the second mating, showing the stably phenotype. I will show you only the main results of this experiment. First, we have the mating rates, which are the dark gray bars, where we can see that the number of mated pairs is reduced at high temperatures, and it's a gradual decrease at higher temperatures. And secondly, as we expected, all the females that mated first with the male grown at high temperatures remated, while females mated first with the control male had a hard time to remate when the stably male was presented, as they were already satisfied with the first mating. Secondly, we have here the proportion of offspring, and this is the offspring with the wild-type phenotype. So you can see that the control group presents a much higher amount of wild-type offspring, while for males grown at high temperatures, it's really low. So females really prefer to store the sperm from the stably male rather than the wild-type. At this point, there are two important points to consider about the egg laying rate, because females could lay eggs, but these do not eclose, indicating that the fertilization is not possible, or females don't lay eggs at all, or they lay a lower amount of eggs, indicating that the seminal fluid proteins are affected by the temperature, as seminal fluid proteins increase the egg laying rate. This plot here shows the number of eggs counted after mating on 3 different days during sexual maturation, and you can see that at higher temperatures the amount of eggs is much lower than the control group. On the other hand, if we look the total offspring produced and the egg to adult survival, we can see that there is a huge increase from the recovering flies, while the males kept at the growth temperature don't really improve over time, but in general it's below the control group. So these results together suggest that both seminal fluid proteins and the sperm cause the reduced fertility and survival. So next, it's important to consider the sperm. It's the second essential factor involved in fertility. And we check for the presence of sperm in the testes, but also in the seminal vesicles, where the sperm accumulates when it's mature. So the mature accumulated sperm and the seminal fluid proteins produced in the accessory glands are transferred together to the female when mating, and both are essential for an optimal fertility. So this plot here shows the sperm presence in the testes, and lines overlap here for the control group and both treatments at 29 degrees. And you can see that there is a much lower amount of the sperm at 31 degrees, so when facing a severe challenge. But if we check the presence of sperm in the seminal vesicles, we can already see the effect of the temperature at mid-challenging temperatures. It's important to highlight that the recovering flies will improve over time in this case, while males kept at the growth temperature don't improve compared with the control group. And also with this example here, you can see that at higher temperatures, the sperm accumulated into the seminal vesicle is null. So there is an error during the spermatogenesis process, avoiding the sperm to maturate. We also measure the seminal vesicle area as a proxy of the sperm amount inside the seminal vesicles, and it also supports our hypothesis showing that at higher temperatures, the seminal vesicle area is much lower, and hence the amount of sperm is also reduced at high temperatures. So we have seen that an error occurs during the sperm maturation phase. So we are now studying the kinetics of spermatogenesis. So here there is a picture of, as an example, showing the abdomen of a Drosophila melanogaster male. And the blue arrow shows the testes, and we will count the number of cells inside the testes following all the spermatogenesis process by comparing what's going on at different temperatures. We have seen that sterility can be caused by a combination of malfunctioning reproductive traits, so inefficient functionality of the male reproductive system given by the accessory glands and seminal fluid proteins, but we have seen also an alteration of the spermatogenesis process. Also, temperature affects differently the male reproductive system depending on the male age. Now I want to thank my group, especially Claudia Fricke, and thank you all for your attention.